Hello everyone, my name is Anup Kumar. Today I am going to present on the topic biological data mining and its application in healthcare under the supervision of Professor M. Sufyan Beg sir and Mr. Misbah Ulaq sir. Let's start with introduction. Data mining has become a crucial research area for life sciences and healthcare applications. Biologists and clinicians are stepping up their effort in unraveling the biological processes that underlie disease pathway in the clinical context. It focuses on the methodologies and process for automatically or semi-automatically extracting useful patterns, insights, or knowledge from large amount of data. However, in order to translate the vast amount of biomedical data into useful insights for clinical and healthcare applications, there are data mining difficulties that have to be overcome, such as handling noisy and incomplete data. Its importance has risen with the rapid growth of online information on the web as well as databases used by industries. For example, various biological or clinical databases result in the increasingly urgent need to derive novel knowledge from the large accounts of data. Objective. At present, there is tremendous growth in biological data, which is primarily due to developments in genomics, proteomics, DNA microarrays, biomedical imaging, biomolecular interactions, and digital records of patients. These data are rapidly increasing, but the tools and techniques to generate the information from this data pool are limited. Using data mining techniques and model automatic discovery of novel patterns is possible from a large amount of biological data, and it has shifted the focus of the researcher towards data intensive discovery. Types of biological databases. First one, human-related database. It decodes the human genome bears great significance in from a theoretical view, unveiling human evolutionary history, and from an application view, exploring personalized medicine against diverse diseases. DNA database. DNA database centers on managing DNA data from many or some specific species. The primary function of human DNA databases includes establishment of the reference genome. RNA database. It is well acknowledged that only a tiny proportion of the human genome is transcribed into mRNAs, whereas the vast majority of the genome is transcribed into dark matter, that is non-coding RNAs that do not encode proteins, including microRNAs and smaller nuclear RNAs. Protein databases. The purpose of constructing protein databases includes collection of universal protein, identification of protein families and domains, reconstruction of phylogenetic trees, and profiling of protein structures. Expression database. Expression database can be used for various purposes, including archiving expression data, detecting differential and baseline expression, exploring tissues specific gene expression and regulation, and profiling expression information based on both RNA and protein data. Pathway database. Pathway databases contain biological pathways for metabolic, signaling, and regulatory pathway. A representative example is CAG pathway, a curated biological pathway resource on the molecular interaction and reaction network. As a core of KEGG pathway integrates many entities that are stored in KEGG sibling databases, including genes, proteins, RNAs, chemical compounds, and chemical reactions. And last, disease database. There are at least 200 forms of cancer in the world, causing 14.6% of all human deaths. Thus, obtaining complete cancer genomes and identifying molecular mutation and abnormal genes can provide new insights for cancer prevention, detection, and eventually personalized treatment. Sequences of data mining. First, data cleaning. Second, data integration. Third, data selection. Fourth, data transformation. Fifth, data mining for knowledge discovery. Sixth, pattern evaluation. Seventh, knowledge presentation. Data mining techniques. Classification, clustering, regression, outer, sequential patterns, prediction, association rules. Let's start with classification. Classification is a data mining function that assigns item in a collection to target categories or classes. Classification is used to accurately predict the target class for each case in the data. Clustering. The purpose of making a group of abstract objects into classes of similar objects is known as clustering. In the process of cluster analysis, the first step is to partition the set of data into groups with the help of data similarity, and the groups are assigned to their respective labels. Regression. Regression is a data mining technique used to predict a range of numeric values. It is also called as continuous values given a particular data set. For example, regression might be used to predict the cost of a product or service given other variables. Outer. Outer detection is a type of data mining technique that refers to observation of data items in the dataset which do not match an expected pattern or expected behavior. This technique can be used in variety of domains such as intrusion, detection, fraud, or fault detection. Sequential patterns. Sequential mining is a topic of data mining concerned with finding statistically relevant patterns between data examples where the values are delivered in a sequence. It is usually presumed that the values are discrete and thus time series mining is closely related, but usually considered a different activity. Association rules. Association rules mining at a basic level involves the use of machine learning models to analyze data for patterns or co-occurrences in a database. It identifies frequent if-then associations, which themselves are the association rules. Organization. First, sequence analysis. Sequence analysis is a term that comprehensively represents computational analysis of a DNA, RNA, or peptide sequence to extract knowledge about its properties, biological function, structure, and evolution. It carry out sequence analysis efficiently. It is important to first understand the source of the data, that is, the different experimental methods used for determining the biological sequence. It needs to follow analytic strategies depending on whether the sequence is genomic, transcriptomic, or proteomic. Databases currently warehousing the enormous data on these biomolecules will need to be first checked for the presence of similar sequences, which might direct experimental assays for functional investigation. Biological network mining. Biological network describes how different biochemical entities interact with one another to perform vital functions in an organism. The ability to determine such network efficiently is important for biological knowledge discovery. Indexing for similarities queries on biological networks discusses three network indexing methods, namely featured based indexing, tree based indexing, and reference based indexing for efficiently accessing and querying these biological networks. It also provides an experimental comparison of applying the three methods for querying gene regulatory networks in terms of running time and accuracy. Third, classification, trend analysis, and 3D medical images. Classification and its application to drug target prediction. We discuss how to apply classification method to a critical problem in a drug discovery pipeline. The predicted drug target interaction may lead to the discovery of new drugs that interact with desired protein target. They are also useful for understanding the possible causes of side effect of existing drugs that could interact with undesired protein targets. Classification, trend analysis, and 3D medical images protein interaction research is useful for predicting protein cellular functions, providing insights into disease mechanism, and developing new drugs to prevent the diseases. Fourth, text mining and its biomedical applications. Text mining in biomedicine and healthcare provides an overview of essential text mining technologies that can be applied in biomedicine and healthcare. It includes entity recognition, entity linking, relation extraction, and co-reference resolution. For demonstration, a case study of a biomedical text mining database system that automatically recognizes and collects cardiovascular disease related genes. Elimination of protein interaction networks is essential for understanding biological processes and mechanisms. Significant information regarding protein interaction networks are available. Conclusion. Biological data mining is becoming increasingly integrated into the entire pipeline of biological and medical discovery processes. There is now an ever pressing need for data mining researchers to collaborate with the biologists and clinical scientists. Data mining researchers have now the opportunity to contribute to the development of the life and clinical sciences by creating novel computational techniques for discovering useful knowledge from large scale real world biomedical data. The ultimate success of the biological data mining for healthcare applications will therefore depend on parallel improvements both in the biological and clinical experimental techniques from the biologists and clinicals to provide rich and clean biological clinical data sets for data mining community. References. These are the references which I have used. Thank you. If you have any query, you can email at anupmu1234@gmail.com.